I'd like to introduce our next speaker. Dr. Chamberlain is the John and Donna Kronecki Professor of Genomics and Personalized Healthcare and Associate Professor of Genetics and Genome Science at the Youth and Health. And an amazing advisor, scientific advisor to the Duke Alliance.
And poker player.
And poker player.
Thank you.
One last
Thank you.
Our poker champion.
All right. And so it was great to follow Matt and Matthias because this is a little bit of the flip side. They told you what happens when you put extra UBE3A in a mouse, and we're asking what happens when you reduce UBE3A from duplication neurons. And so it's kind of a great juxtaposition of these 2 points. So I'd like to tell you a little bit. So most of my talk today is going to be on how we've developed these models, because my talk is also a setup for a talk tomorrow from Eric Levine's lab. And if I don't say it enough during my talk, make sure you listen to his talk, because he has some really cool data that I think is pretty important. So these are the disclosures I'm required to tell you about. I just really have to point out the financial conflict of interest to be on the up and up. So my lab studies genomic imprinting disorders that are caused by copy number variation on chromosome 15, Q11 to Q13. There are 3 disorders: Prader-Willi syndrome, Angelman syndrome, and Du15q syndrome. And because we study gene regulation, we find that it's important to understand all 3 of these disorders. They help us understand what's going on the maternal allele, what's going on the paternal allele, and then what happens when you have too many copies. And so it's been really important that we study all 3 disorders. So you'll hear me talk about especially Angelman syndrome a little bit today. So you'll recognize this figure. This is the genetic region on chromosome 15, Q11 to Q13, and it's why the parent of origin is important for this disorder. So the genes here are regulated by a process known as genomic imprinting, and that means they're made exclusively from either the paternal allele or the maternal allele. So the genes shown in blue are made exclusively from dad's copy of chromosome 15. And the one gene in red here, UBE3A, is made exclusively from mom's copy of the chromosome in neurons. And I want to point out, if you're missing this region on the paternal allele, you'd have Prader-Willi syndrome. If you're missing it on maternal allele, you would have Angelman syndrome. And these deletions and duplications are mediated by these breakpoints. And you probably heard Dr. Shannon earlier talk about those breakpoints and those being repeat regions where the chromosomes misalign during meiosis. And so those same misalignments that cause deletions can cause the duplications. So my lab studies these disorders. So I want to say that, first of all, that map is pretty complicated, right? There's a lot of genes and it looks quite complex. And so how do you model this? And Matt and Matthias pointed out like how arduous it might be to do this in the mouse, to go gene by gene. But in humans, we can use something called patient-derived iPS cells. And what this means is that we can take a sample from an individual. It used to be like a lot of the cell lines I'll talk about today were derived using fibroblasts, which means we got them from a skin punch biopsy. But now we prefer to do this from a blood sample. And so we can take those cells and we can convert them, we reprogram them into stem cells. And the reason we want to do this is those stem cells can be maintained in culture indefinitely in the lab. We can make more of them, we can grow up tons of them, and we can freeze them down and save them forever. And importantly, they have the exact same genetic change that that patient sample we started with had. Okay, and so it's really a way to preserve the genetics in these stem cells. And on top of that, we can take these stem cells and using a variety of different strategies, depending on the question we're asking, we can turn them into neurons. Technically, we could turn them into almost any cell type in the human body, but we're interested in neurons because that's the affected cell in your kids. And so we can convert these into neurons, and but we don't want to do this to put the neurons back into your kids. That's not a good idea. Your kids, their brains are largely functionally and structurally intact, and frankly, we don't know how to reintegrate them very well with a lot of fidelity. And so instead, we want to use these iPS-derived neurons as disease models to better understand the disorder, but also to use them for drug discovery. And I hope I'm going to be able to make a case for you today of why and how this is possible. So, this is a bit of a review too, because Dr. Dewis and Carolyn Shannon told you about this genetics, but I want to quickly point out that, most individuals, all of us, have a copy of chromosome 15 from dad and a chromosome 15 from mom. UBE3A is made only from mom's copy. Angelman syndrome has a deletion on the maternal allele. Prader-Willi has a deletion on the paternal. Do15q is a little bit complicated, right? There's a lot of different flavors of this. And I'm going to talk primarily about the idic instances, the isodicentric ones here. But there are also interstitial duplications and interstitial triplications, as well as paternal interstitial duplications, albeit it's a slightly different disorder. And so we have made IPS lines from 3 different individuals with idic 15. We have one from a maternal interstitial triplication, one from a maternal interstitial duplication, and one paternal interstitial duplication. On top of that, we have Angelman and Prader-Willi and typical control cell lines. And so we have a really nice toolkit to start asking questions. One of the questions that you've heard is important to ask is the parent of origin. Can we confirm that these idics or duplications are maternally inherited? And we turn to the DNA methylation test that many of your kids may have had upon their diagnosis to ask the question, in our stem cells, do they retain the identity of being maternal? Okay, and we think that that's important to do in case it gets erased. Thus far, we've never seen this be erased in Do15q cells. We've never seen it be erased in Prader-Willi or Angelman cells either. So it's really maintained tightly. But importantly, and this will become important later in the talk, our idic cells have 75% DNA methylation. So you and I sitting in here all have one paternal allele and one maternal allele. Dad's copy's unmethylated, mom's copy's methylated. And so we have 50% methylation, right? 50% methylated, 50% unmethylated. Kids with an idic chromosome have 3 maternal copies, and that's why they're 75% methylated. 3 maternal, 3 methylated, 1 unmethylated, okay? And so this is maintained nicely. So we've established that we can make these cells, and we've established that they retain the parent of origin identity that's necessary to study them. So can we use these cells to help develop therapeutics? So I'm going to turn to a different disorder we study to help demonstrate proof of concept. So first of all, my lab also studies Angelman syndrome, and I'm going to say this differently than I ever would in an Angelman syndrome talk. I'm going to say that Angelman syndrome is a contiguous gene syndrome, meaning multiple genes caused by deletions on maternal 15q11 to q13. And that's an accurate statement. 70% of the kids with Angelman syndrome have this large deletion. And if you were to look at the chromosomal region again, kids with Angelman syndrome have this maternal deletion. Not only are they missing the maternal copy of UBE3A, but they're also missing 1 copy of all of these other genes shown in gray. Okay, so they really are missing additional genes. And in fact, the kids with deletions, with Angelman syndrome due to deletion, have a more severe phenotype than Angelman syndrome due to different genetic etiologies. And in fact, we've used these other genetic etiologies, specifically UBE3A mutations, to really tell us that UBE3A is the driver gene in Angelman syndrome. It is the gene that is probably the most impactful for Angelman syndrome. That doesn't mean that the other genes aren't contributing, because as I just told you, the kids with deletions are more severely impacted than those with UBE3A mutations and the other genotypes as well. But to say that UBE3A is the driver, it's the most important. So with that in mind, I have to tell you about a few more tools. So we know UBE3A is the driver gene for Angelman syndrome. We use these tools called CRISPRs to help make cellular models that help us test things better and to help make our cellular models better. CRISPR, what it stands for is probably irrelevant. What you need to know about it is it's a set of DNA scissors. It's comprised of 2 pieces. Cas9 is a protein that's shown here in yellow, and it cuts DNA on both strands. And then the other piece of it is a guide RNA, this green squiggle here, that directs the scissors to one specific DNA base pair in the genome. And so what happens is it makes this cut. Why you would want to do this is so that you can either change or remove pieces of the DNA. So I have to pause here and tell you, I think this is a terrible idea therapeutically for Du15q syndrome. CRISPRs are in the news a lot as a therapeutic approach. I just don't think they'd be terribly helpful for the reasons I talk about here. So what happens after the cut? Well, most frequently, this process called non-homologous end joining, it doesn't matter what it's called, it's the process by which this cut is repaired. Cells remove DNA on both sides of the cut a little bit and then try to fill it in. And that process is really error prone. And so that's why, so if you were to use a CRISPR in your child and cut the DNA, it's going to make a mistake. And in fact, we can't predict what those mistakes are. And 2/3 of the time, it isn't going to disrupt the gene. It's not going to make an error that changes the protein if that's what you intended to do. So it's an uncontrollable error, in fact. Secondly, the second thing that happens, and it's somewhat rare, in our hands, it ends up being about 10% of the cases when we're trying to do this. If we use 2 CRISPRs, those 2 cut sites, instead of making that error-prone fill-in, will join together. And these cut ends join and you can delete something that way. Again, it happens in about 10% of the cells that got the CRISPR in our hands. And then the third thing that happens, even more rare, it happens about 5% of the time when we try to accomplish this. We can use a template. And by doing that, we stick in a copy of DNA, a small piece of DNA that we want copied into that place we just cut. Again, 5%, that's pretty inefficient at the end of the day, and that's in a dish in the lab, yada, yada. So why would we do this? Well, we do this in Angelman syndrome to develop isogenic pairs of stem cells. What's isogenic? Iso means the same, genic means genes. These are identical twin stem cells with and without the disorder. And the reason we want to do this is because when we look at a phenotype in a cell, we don't know whether it's due to normal human variation or whether it's due to the disorder we're trying to study. So in this top example, in the middle example here, this is a case where we've taken stem cell from an Angelman syndrome patient that had a single base pair change in UBE3A. It was TCT when it needed to be TTT. We used CRISPR and a repair template to change the TCT to a TTT. And we know it was a change and not a mix up in the lab because we put a little passenger mutation here that helps us track that it was an edited cell line and not a unintentional mistake. We can also do this by deleting UBE3A. So in this case, we use 2 CRISPRs and use that end joining part, right, where the 2 cut ends recombine to each other, just to delete UBE3A. And then after the fact, asked whether we cut the maternal copy or the paternal copy. So in this way, so why would we do this? We do this because, again, we can more easily identify the phenotypes and trust that they're really due to the disorder, trust that UBE3A caused them in this case. And then, down the road, this gives us a more confident testing for potential therapeutics for the exact same reasons. We can be certain that it's restored the phenotype and not just due to genetic variation in that patient-derived cell line. So we can also generate isogenic mutations for the larger deletions too. And in this case, we used CRISPRs targeting those repeats. So remember, you heard earlier that at each of those repeats, breakpoint 1, breakpoint 2, breakpoint 3, breakpoint 4, those are the same breakpoints that you might have heard in your child's diagnosis. We targeted a CRISPR to those, and it cuts multiple times at each of these breakpoints. And so we stuck the CRISPR in. It's going to cut a lot of places. Most of the time, it doesn't do anything that we can detect. It could be making huge mistakes for all we know. But we can go through and we can screen for UBE3A expression. In stem cells, UBE3A is made from both parental chromosomes. And so if we asked which cells had a little bit less UBE3A, we could hypothesize or guess that they probably had a deletion between breakpoint 2 and breakpoint 3. Or actually, I should say, it just included the region between breakpoint 2 and breakpoint 3, but it could have involved breakpoint 1 and breakpoint 4 and so on and so forth. So once we have these deletions, we can then ask which parent did it come from? So we go back to this methylation test. And we identified one cell line that had a Prader-Willi deletion because the remaining copy was 100% methylated. It was a methylated maternal allele. The other case was we had two Angelman cell lines. This is the example of one that's completely unmethylated. They were of different sizes. They used different breakpoints. The Angelman one was 5.8 megabases, million base pairs. And then we did some gene expression to confirm that they really were Prader-Willi-like and really were Angelman-like. And so in this way, we could make identical twin cell pairs that had those larger deletions. So can we do this for idic chromosomes? We weren't the people that invented this idea. We just borrowed the idea and tilted it to idic 15. So what these guys did was they took advantage of repetitive elements that are specific to individual chromosomes and asked, could you use those to get rid of whole chromosomes? And they did this to get rid of a Y chromosome, because who needs more men, right? Kidding. But they also did it to remove an extra chromosome 21 in trisomy 21 cells. So we tried this approach. So we started with our CRISPR that cuts in the Golgi 8 repeats, these repeats. And so I just want to point out quickly that this is an idic chromosome. Those two black circles represent the centromeres, right? The dicentric pieces. And so there are actually, many sites for that CRISPR on an idic chromosome. On top of that, we added an extra shredder. We designed CRISPRs to cut in these other repeats that are affectionately known as SNORD116 and SNORD115. We wanted to shred this chromosome. We did that, and instead of screening for UBE3A expression, we screened for copy number. We asked how many copies of UBE3A are there in the cells that got these CRISPRs. And what we found was that 4 out of 58 had copy number changes in UBE3A. And thankfully, we had controls because we'd made these cell lines. We have H9 as a neurotypical cell line. We had a maternal interstitial dupe that had 3 copies of UBE3A, and we had idic 15 with 4 copies of UBE3A that allowed us to ask how many copies did our clones have. Our clone 5, and clone just means one cell that we captured that had these mutations. So clone 5 and clone 6 here have a pretty substantial reduction of UBE3A. We then asked which parent did it come from. I'm going to remind you about this DNA methylation assay, where we're expecting the idic cells to have 75% DNA methylation. Remember, because they have 3 maternal copies and 1 paternal copy. And if we got rid of that idic chromosome, we would expect a 50% methylation, right? Because we got rid of those 2 extra maternal copies. And much to our delight, we got one of our clones, clone 5, had 50% methylation, suggesting it had lost the idic chromosome. So what happened to the others? So, our output was 1 out of 58 did the right thing. So that's reason number 500 not to use CRISPR as a therapeutic approach for this disorder, at least DNA cutting CRISPRs. The other 4 that had copy number changes, one of them we know probably lost the paternal allele. Everything that's remaining is 100% methylated, so we lost the paternal allele. Clone 6 looks like it lost one of the maternal copies. We don't know if this was on the idic chromosome or if this is on the parent chromosome. Let's see, what was the other one? Clone number 18 also looks like it lost the paternal chromosome because it's almost 100% methylated. So we then took clone 5 and asked a little bit further. We did a chromosomal microarray, which is again a genetic test that your kids might have gotten along the way to ask about copy number changes. And so our unedited duplication idic cell line actually has a few different copy number changes, as do most of us, right? Most of them are benign and they don't do much, but it has this 15q11 to q13 gain that they picked up. In our corrected dupe clone, they had all of the other genetic changes, the deletions on chromosome 5. I can't remember what all's in these. There's a loss of heterozygosity on the X chromosome, but importantly, they no longer had the 15q11 to q13 gain. And so that told us that it did change the copy number in the way we'd expected. And finally, we did a karyotype. And so I didn't do this on purpose, but I feel like I mirrored what Carolyn Shannon and Jessica Dewis told you guys earlier about how you go through the diagnostic steps, right? We do this with our cell lines to make sure we have what we think we have too. So these guys, you know, when we edited, we went from this extra little chromosome having the extra idic 15 to having no idic 15 and no other observable changes. So we have identical twin cells with and without the idic chromosome. So how can we use them for drug discovery? So I need to tell you about one other tool. These are antisense oligonucleotides. You may have heard of the drug Spinraza. Has anybody heard of that? It was an outstanding therapeutic for a disorder called spinal muscular atrophy. And what that drug did is it's a little piece of DNA that, can come in and modulate the splicing of how RNA is cut and put together. Okay, and that's how Spinraza works. But these antisense oligos, or ASOs, also can do another thing. They can cut the RNA and degrade it. And that's how you can lower gene expression. We love these in the lab. They're wonderful tools because they can knock down a gene, whatever gene we choose. And they do so really specifically. They don't change other genes in the genome. They just change the one we've asked them to. And importantly, these ASOs also are freely uptaken by neurons. We just dump them in the media and neurons suck them up and they do their thing. So again, for Angelman syndrome, why are we not moving? Ah, there we go. Okay, yeah, so for Angelman syndrome, remember that I told you earlier that they're missing maternal copy of UBE3A. Dad's copy of UBE3A is silenced. It's silenced by a long non-coding RNA, this thing called the antisense. That's what this dotted line means. And so ASOs, a lot of different companies and a lot of different labs have figured out ways to cut this RNA and turn back on Dad's copy of UBE3A as a therapeutic approach. And so our cells have been instrumental in helping develop these therapeutics. This is some of the work we did with Ionis Therapeutics. An ASO that's targeted against the antisense can reduce the antisense and therefore increase UBE3A. And what's cool about this is the ASOs work in a dose-dependent manner. If we didn't want to knock all of it out, we could add less ASO and you get a lesser reduction. This is important for something like DUP15Q where we need to leave some of the genes there, right? We don't want to get rid of all of it. We want to get rid of some of it. What's cool also about this particular tool, these ASOs, we could add them once to the neurons in a dish and then come back and ask 7 weeks later, we just feed the neurons as normal for 7 weeks. And you still see the same reduction in UBE in the antisense. So they're long lived. They last for a very long time in the cells, which is really cool. We also know that, so we asked whether these ASOs, when we put UBE3A back, do they rescue the cellular phenotype? What goes wrong in the neurons with Angelman syndrome? And so one of the most obvious phenotypes for Angelman syndrome was found by James Fink, who's sitting here. James found that Angelman syndrome neurons have a more depolarized resting membrane potential. They're less negative. And when we added the ASO to our Angelman neurons, it restored the negativity in the neurons. I don't mean that in a bad way. This is when negative is good. They're more hyperpolarized. And there were other physiological phenotypes that I won't talk about here, but this was really the main one. Is it changed this resting membrane potential change, and as a result, Angelman cells are slower to mature. Their action potential firing patterns are slower to mature. And our ASO also rescued that. It rescued the maturation of the action potential firing patterns. And so here, and so now this, not this ASO, but an ASO like it is in development as an Angelman syndrome therapeutic, and it will be in clinical trials, they tell me, within the year. So, okay. We have UBE3A as a driver gene for Angelman syndrome. We showed you that ASOs can work for this. So what's the driver gene for DUP15Q syndrome? And you just heard that maybe it's not UBE3A, or maybe UBE3A is questionable. And and this slide is not meant to tell you any useful data other than to say, again, it's complicated. There's a lot of different genes, and there's a lot of different gene dosages. We have a bunch of different cells here that are maternal interstitial dupes, paternal interstitial dupes, and idics. And there's not a clear winner here in the candidate gene race. However, we can take a bit of advice from human genetics, most notably the interstitial duplication folks. Maternal interstitial duplications are every kid who has one, I think, has Du15q syndrome, but very rarely do kids with paternal interstitial duplications have the syndrome. They have a different disorder. And in fact, I met a very cool young lady, and her mom was an example of a maternal interstitial dupe that was inherited from a mom who also carried it paternally, and they were very different. And so, so the only difference between these is really UBE3A. The same genes are duplicated except for UBE3A, and UBE3A is extra in these maternal interstitial duplications. And there's one other piece of evidence. We know that there's this one family, at least, that's been published that has a duplication of just UBE3A, and it's a multi-generational family with multiple members that have the duplication, and every member who has the duplication of UBE3A has developmental delay and neuropsychiatric phenotypes. Mind you, it's not Du15q, but it's developmental delay and maybe some of the same features. So, I mean, I think that this brings up a possibility that UBE3A may indeed be a driver gene. It's necessary for Du15q syndrome, but it's not sufficient. UBE3A duplications alone aren't sufficient to cause 15q duplication syndrome. So what do we do with that information? And like Matt and Mateus talked about, you test the hypothesis. You ask the question of what happens now when we reduce UBE3A. And I'm not going to give you a lot of answers. I'm going to say, you know, if you want to know the answer to that question, come to Eric Levine's talk tomorrow. I'll give you a little bit of a flavor though. Oh, and I need to mention one other set of tools we have are other genes driver genes. So we have to make the tools to do this. And so my lab has also designed antisense oligos against, so I showed you the UBE3A, no, I didn't. We've designed ASOs against UBE3A and found ASOs that reduce them to normal levels. We know the dose that is needed to reduce them to typical levels. We found ASOs that can reduce HERC2 to typical levels and GABA beta 3. We still can't find one for GABA alpha 5. For whatever reason is a little bit intractable to reduce it. But nonetheless, we have all these tools in the lab now to ask the question of which genes are the driver genes. And I also want to mention that there's an untargeted therapeutic approach too. We stumbled upon this drug mithramycin. It was a suggestion of a colleague of mine, Mark Lalonde, who suggested, why don't you try this? It's supposed to reduce gene expression. And we tried it on UBE3A, and in a dose-dependent manner, it reduces UBE3A, and there's kind of a max out effect. It can't really lower it all the way to 0. It just reduces it, which we were quite excited about. And we haven't followed it up because, you know, as a geneticist, it's not targeted. It probably changes the expression of other genes, but we're revisiting this and like, I think we have to keep an open mind that maybe it is worth following up and seeing whether this would be a good approach. So nonetheless, you know, Eric's talk is going to do a better job of explaining what happens when we reduce UBE3A in our stem cell models. And it's a complicated story too. And it's the work done by Marwa, who's sitting next to James here. So if you want to preview, track Marwa down. I'll give you a tantalizing bit of data though. So when we reduced UBE3A in our idic cells using an ASO and asked about morphological phenotypes. So Du15q neurons have a smaller soma size and have an increased density of dendritic protrusions, these little blubs off of the neuron that eventually become dendritic spines. So when we added the ASO, we can first of all normalize the UBE3A levels, like I mentioned. So the purple here are idic 15 cells, blue are the neurotypical cells, and pink are the ASO treated cells, okay? This just shows you the UBE3A levels after ASO treatment. The middle graph shows you what happens when we measured the soma size, and this is the work of a collaborator, Les Lowe, and his student Judy Bloom. In purple, again, the idic cells have a smaller soma size. That's the piece of the neuron where the nucleus is. It's the body of the neuron, if you will. When we add the ASO, we can normalize that. It's closer to the neurotypical neurons. And the dendritic protrusion density is increased in idic 15 cells at different stages of development. And when we, again, normalize UBE3A using the ASOs, we can restore those to normal levels. And so I think that this is, and like I said, Eric's data is, Eric and Marwa's data is much richer, and they'll tell the story more fully. But we think that decreasing UBE3A may very well be a promising therapeutic approach for DUP15Q, and we think it's worthwhile following it up. So with that, I need to thank the people who did the work. The cell lines that I talked about, the engineered cell lines for DUP15Q were made by Chris Stoddard and a summer student visiting from France, Aurélie Dumarche. Dea Gorka and Noel Germain did a lot of the gene expression and the ASO work initially that I talked about. I have important collaborators. I couldn't do half of what we do without collaboration. Eric Levine and Les Lowe at UConn Health, and our Frank Rigo at Ionis helped us with the ASOs. We have a fabulous cell and genome engineering core. I have to thank the families who donated samples. Without you guys, we couldn't do this research. And also our funding sources, the Angelman Syndrome Foundation, the NIH, and the Connecticut State Stem Cell Group. With that, I'll take any questions if there's time.
Well, we got Ron on the line too.
Oh, we don't need to listen to Ron.
But we don't need to see Ron. He can hear you.
Oh, we need to listen to Ron.
Oh, and biosamples. If you are willing to give a biosample
That's what I thought you said, Stormy.
for this work, please do sign up to do it. We do have a bottom here, some plastic for families. We are doing full quads and trios. How many ever people want to donate? We're able to do it here. So, what? I'm not surprised. I connected with Matt. Met the both Matt.
Is there any reason to be discouraged by the mouse models versus the iPSCs in terms of the phenotypes?
No, I don't think so, because I think, I mean, frankly, I think we need to proceed with caution, right? And as a scientist, I always remain skeptical, right? But I think it's this necessary versus sufficient bit, right? And they kind of alluded to it too. It might be UBE3A plus another gene, but if UBE3A, and I think maybe there's an important distinction here too. You can't expect it to cure, right? If you put UBE3A back, even if you put it back reduced it appropriately. Even if you did that very early in development, you can't expect it to cure the disorder because there are other genes, right? But will it make a substantial benefit? I think it's worth asking the question. So I'm not discouraged at all. I think it reminds us to be cautious.
Any other questions for Stormy right now?
Oh, Matt's got his hand raised.
Matt wants to respond.
Yeah, it's just, it was a great talk, Stormy. I really appreciated it, and I hope everyone else did too. I just had a quick question. Considering the, let's see, like biallelically expressing cell types, and when UB3A overexpression might be influencing those, say at very early stages of brain development, do you think that might impact the treatment window and maybe shift it a little earlier, or what have you, when you consider treating this versus something like Angelman syndrome with something like an ASO?
Yeah, no, it's a great question. And it's a tough question, right? As you, I'm sure know, we can't answer that. So what Matt's alluding to is dad's copy of UBE3A gets shut off during neuronal maturation. And so there's this period of time in neural progenitors and in early neurons where there's actually all 4 copies of UBE3A are expressed in idic kids. And so does that mean, is it that early expression, is that early extra expression, is that going to make it harder to treat therapeutically, because maybe that's the window you need to hit it at, right? When there's 4 copies and not 3 copies expressed. And so this is one of the tough parts about the mouse models being difficult, right? Is that, you know, for the Angelman mouse, Ipe's lab, Ipe Algarisma's lab was able to ask, when do you have to put UBE3A back in order to rescue phenotypes? And they have phenotypes to rescue. And we thus far can't do that in the mouse. And so I'm hoping that, you know, you guys are able to mate your duplication mice to the larger duplication and see whether putting extra UBE3A in the context of the other duplicated genes would be helpful as a mouse model where you could very much ask that question. And I think you guys would be able to do it better than I in this case.
What?
Anything else?